Okay, let me first thank the organizers of this symposium for giving me the opportunity to share with you some of the results of the Belgium study. The Belgium study is a study with quite broad scope. Its general aim is to investigate the complex relation between aging, health, disability and frailty in a multi-system approach. Today I will focus on the immune system and more specifically the effects of CMV on the immune system and its consequences in the oldest old. Maybe I should apologize in advance. There will be some overlap with the presentation by Graham. I saw that we even have the same graphs, pictures, tables, but that give me the opportunity to go through a bit faster, to go a bit faster through some of the slides. Okay. Very briefly, immunosenescence. Of course, all components of the immune system are affected with aging, but hallmarks are inflammation, which is a chronic low-grade state of inflammation, and that has been shown to contribute to the development of many age-related diseases. The second hallmark is T-cell senescence, which has this has been said before, I think, which is characterized by an accumulation of late differentiated T cells. T-cell senescence affects all T cells, but is most visible in the CD8 compartment. And the most obvious consequences of T-cell senescence are, as we already said, increased susceptibility to infectious diseases, decreased response to vaccination. It should be noted that for long, immunosenescence has been regarded as an essentially detrimental process, whereas recent insights suggest that some of these changes might in fact be appropriate adaptive changes rather than just a decline of the immune system. Cytomegalovirus belongs to the herpes viridae family, is highly prevalent all over the world, within some regions of the world, prevalence rates up to 100%. For long, a primary CMV infection establishes persistent infection, and for long, persistent CMV infection has been regarded as relatively harmless in immunocompetent persons. But in recent years, a growing body of evidence suggests that CMV infection might be implicated in the development of many chronic diseases, and I think the best example is atherosclerosis. But CMV infection has also been implicated in cognitive impairment, physical impairment in elderly and in immunosenescence. The first, the Swedish Octo and Nonna studies were among the first to indicate that CMV might play a role in the process of immunosenescence. Based on these studies, an immune risk profile was defined. This is a slide that Graham showed you too, consisting of a set of parameters, being expansion of CD8 late differentiated T cells, support T cell proliferative response, low level of B cells, an inverted CD4-CD8 ratio, and CMV seropositivity. This IRP was associated with increased risk of mortality at 2, 4, and 6 years follow-up. And it was furthermore shown that the CD4-CD8 ratio alone could be used as a proxy for the complete IRP to predict mortality. Further studies then confirmed that indeed CMV infection is associated with an accumulation of these late differentiated CD8 T cells, and furthermore it was shown that many of these late differentiated CD8 T cells were CMV specific, suggesting that that it takes a large effort of the immune system to contain the CMV infection. In last 3 years, 2 large studies have been published regarding the association between CMV infection and mortality, one in the UK and one in the US. This is a Kaplan-Meier curve from the study in the US, and it shows survival in function for different permutations of CMV serology status and CRP levels. And it clearly shows that survival is shorter among people with CMV infection and high CRP levels. The mechanisms by which CMV impacts on the immune system are far from fully understood. A common hypothesis is that it may impact through repeated cycles of reactivation with concomitant inflammatory reaction and oligoclonal expansion of CMV-specific T cells. It is believed that reactivation of CMV infection occurs more often with aging, but I must emphasize that this is based on the observation that anti-CMV IgG titers tend to be higher or tend to increase with aging on the one hand, and the hypothesis that the height of the titer is related or reflects the cumulative frequency of reactivation. Okay, this was the introduction, brings me to the Belfrail study. Very briefly, the Belfrail study is an ongoing prospective observational cohort study in community-dwelling individuals aged 80 or older. At baseline, 567 individuals, of whom 63% women, with a mean age of 85 years were included. A comprehensive assessment was done at baseline and repeated approximately 18 months later. And at the latter occasion, we isolated PPMCs from 276 participants for T-cell phenotyping. Thereafter, yearly follow-up was organized in order to collect data on new mobilities, hospitalizations, and mortality. CMV serostatus in the Belfrail study, 76% were seropositive for anti-CMV IgG, which is pretty low compared to many other cohorts of elderly, but which provided us a very good discriminative power. Among the CMV seropositives, 39% had a high anti-CMV IgG titer, which was defined as a titer equal or above 250 international units per milliliter. The titer significantly increased with age and was higher among women than men. T-cell phenotyping showed that in 7% of the Belfrail cohort showed an inverted ratio CD4-CD8 ratio in 7% of the well-frail cohort, which was approximately half the prevalence that was found in the octonona studies. And as expected, this inverted ratio was significantly associated with CMV infection. But to our surprise, we found that about 1/3 of the cohort had a high CD4-CD8 titer above 5, and this high ratio was associated with a low proportion of CD8 T cells when compared to the group with an inverted ratio, but also when compared to the group with a intermediate ratio. We also had a small control group of young individuals, and this high ratio was not found in this group. But because of the fact that this group was so small, we pooled our data with data kindly provided by colleagues of the University of Antwerp. And we pulled them in this graph, and you can see that a CD4-CD8 ratio above 5 does not occur before the age of 50. And second, that the CD4-CD8 ratio tends to increase with age. We then, out of curiosity, went back to the octonona studies, and we found that they also, in about 20% of the population, found CD4-CD8 ratios between 4 and 11, but they didn't pay much attention to this group. Further phenotyping showed that the title is expression of naive, but I should say, I think, naive with a naive phenotype. So expression of T cells with a naive phenotype in function of the CD4-CD8 ratio and in function of CMV serostatus showed that a ratio above 5 was significantly associated with a relatively high proportion of T cells with naive phenotype in the CD4 compartment as well as in the CD8 compartment, and both for CMV negatives and seropositives. With regards to the late differentiated T cells, the picture is a bit different. We saw that, as expected, an inverted CD4-CD8 ratio was significantly associated with a higher proportion of these late differentiated T cells when compared to the group with a high ratio, but this was only the case in the CMV positives. So it seems that we identified a phenotype characterized by a high CD4-CD8 ratio as a result of a relatively high proportion of T cells with naive phenotype as compared to the groups with lower CD4-CD8 ratios. The question is this clinically relevant? And that brings me to the next part of this presentation. What we did, we looked at functional performance in these oldest old, in function and frailty, in function of CMV serology and the CD4-CD8 ratio. These are the results or summarize the relation between CMV and functional measures. As functional measures, we had the MMSE for cognitive functioning, the short physical performance test battery, and activities of daily living for physical assessment, and frailty was assessed by means of the Fried criteria. In contrast to many other previous studies, we could not establish a relation between CMV infection and physical or functional impairment. On the contrary, we found that in this cohort of oldest old, CMV infection was protective against frailty. We also looked at, among the CMV seropositives, at the relation between titer, anti-CMV titer, and functional performance. And again, in contrast to previous findings, no relation between titers, height of titer, and functional performance. We found a relation in the bivariate analysis, but this association disappeared in the adjusted models. I already mentioned it, but it should be emphasized that the BEL-frail cohort was considerably older than the populations in the previous studies. So probably these results, or these contradicting results, are the result of a survival effect. Okay, then we looked at CMV serology, ratio, and functional performance, and this gave a slightly different picture. This graph shows you physical performance for the different groups, and you see that the group with a high ratio, proportion of physical impairment in the group with a high ratio is significantly higher than in the group with an inverted ratio. Then we stratified for CMV serology, and the association remained, but only in the CMV seropositives. Furthermore, we looked at CMV serology and mortality. And this is a Cox regression plot. It's not a Kaplan-Meier. Cox regression plot, again, for different permutations of CMV serology and CRP level. And it shows you that there is there is indeed a relation between survival, duration of survival, and CRP level. The one with the highest CRP levels have the shortest survival, but there is no relation between CMV serology and survival in this population of the oldest old. We also looked at the relation between mortality and anti-CMV IgG titer, and this showed something else. There was a relation between so people with a higher, with the highest anti-CMV IgG titers tend to survive. Die earlier. We tried to analyze the mortality data also in function of the CD4-CD8 ratio. Unfortunately, the sample size was too small, and especially the number of events was too low. So we'll have to wait to do this analysis for the 5-year mortality data, which will soon be available. So, in conclusion, The oldest all seem to represent a phenotype that is less susceptible for the detrimental effects of CMV. And this is probably the effect of the result of a survival effect. But even amongst these survivors of CMV, there seems to exist heterogeneity in terms of the immune response or response of the immune system and its subsequent consequences. In the very elderly, a result suggests that in this group in this population of the very elderly, an inverted CD4-CD8 T-cell ratio may be associated with a more adapted, more advantageous immune response, and as a result, better outcomes in terms of functional performance. I thank you for your attention.
Thank you very much. Sven, shall we go to coffee?
Yes.
Well, that's a question. I want to ask.
Yes, please.
Do my question.
We have, I think we are relatively well in time, so we can have questions.
All right, nobody's away.
Simple question. For these individuals with the really high CD4 to CD8 ratio, what are the absolute numbers? Saying like, cells per milliliter. Are you seeing a collapse of CD8 or what?
I can't answer that question. I have to look that up. I don't know.
Okay, because I mean, presumably it's really important whether it's the CD4 population growing really fast or the CD8 population collapsing. If the CD8 population is collapsing, that might cause all manner of different problems that were...
I would predict whether it is a collapse of the CD8.
It is, yeah, that's it. There is, but it's all in relative terms. It's a relatively, it's an accumulation. Wait, what was the question?
I said, is the ratio so high because you have
No,
an
there is
explosion
a
of CD4?
decrease of the CD8.
Okay,
yeah, okay, sorry. Yeah. Decrease, yeah.
So I think your data on the CMV and I said frailty, that it really doesn't impact frailty, is very...
On the contrary.
Yeah, so it even protects me.
Yeah.
But I think, and also the mortality, I think there's data also from the Leuven group showing that
yes,
relation.
I forgot to mention, there are two studies that have kind of similar findings. Leuven 85 showed that survival was better among these people with low CD4, low naive T-cell counts. And Newcastle 85 found a relation between, found that frailty was less likely in people with a high CD4-CD8 ratio. But that's all there is with respect to...
But the CMV status, I mean, is it now true that it is really a negative effect on health? Because I am not convinced by the data. So
yeah, I personally, because this was just, okay, this study is about what does CMV do, and we were convinced that there is heterogeneity there, in terms of how the virus coexists with the host. Personally, I think CMV, the effects of CMV will depend not only on the host and its immunoresponse, but also on the moment in life. There are some studies that suggest that CMV might be beneficial in early lifetime because it has kind of a bystander effect that might protect or at least help to protect against other infectious diseases. There is a very recent study where it has been shown to increase pneumococcal vaccination response, but we cannot deny the mortality data in these large population-based studies. But I must say that in these studies, highest mortality linked with CMV was found in middle-aged or young elderly. So I think the effect is very diverse.
I had a question. I prefer to keep asking the question because that's exactly the following, and that's very general for CMV. We have a few experts here. So it seems that no one disagrees with the fact that in adulthood, CMV is bad, to make it simple.
It's not for everybody.
Well, bad or not bad, but good.
No, okay.
So there is always the question of whether we should try antivirals. And I hear that for now there is no point to accept antivirals and against CMV. Did I hear right? Or I mean, what is the step to go to humans? Because we've been talking about mice, about animals and humans, and now we're facing an obvious problem in people, and we have some tools, but we don't know what to do. We have some technique. Okay, yes, we agree. Antivirals have some issues, but what are the next steps? How can we go forward instead of having another third, fourth, 10 times such studies? They all show more or less the same. We can discuss in the details, but the big aspect is quite obvious.
I do not agree that they all show the same. As I said, it depends. And personally, I think it's way too soon to start with intervention. I think our knowledge of CMV is just not good enough.
I agree with that completely, because since I stepped into this field, I'm trying to understand why there is such a big course of papers on CMV. We do not have a conclusion yet.
No, but it's fascinating.
Yeah, it's fascinating.
It's so difficult.
So it's hard to understand if it does anything, and yeah. So it does impact the immune system, that's true. I mean, we see effects, and we see antibodies, of course. But it's kind of hard, because the studies are so contradictory.
Do we need to understand? I mean, if the mortality is higher,
It's not always the case.
Not always, not in the oldest old. And as you heard already, this population of 80 plus is growing. So for them,
or maybe they don't take the medicine. But young people, young adults, should take there should be clinical trials with young persons. There it's clear.
I think it's too early.
Okay, thank you very much.